Hey, listeners of the Bio Report. Before we get started this week, I wanted to tell you about the digital library from Deep Dive. How much time does your team spend looking for research papers? Google, PubMed, social media. There's got to be a better way. You can now search a reference database of 100 million scientific papers and read the full text of 20 million articles, annotate them, and share with colleagues. It's the smarter way to do research. Here's the best part. If you're like me and been frustrated by not being able to access articles you find because they're behind a paywall, I've got good news. With Deep Dive, you get one-stop affordable research. If you're a listener of the Bio Report, you can try the enterprise version of the service for free for 1 month. Go to deepdive.com/podcast and enter the code BioReport. That's Deep Dive, DeepDive.com/podcast, and the code is BioReport, one word, all caps. I'm Daniel Levine, and this is the Bio Report. Wet age-related macular degeneration is a progressive disease and a leading cause of vision loss in patients over the age of 60. Vision loss is caused by the leakage of blood and other fluid from abnormal blood vessel growth underneath and into the retina. Though the condition is treated by a class of therapies known as VEGF inhibitors, these biologics need to be repeatedly injected into patients' eyes, and for a variety of reasons, patients' real-world experience with the drugs don't match clinical trial outcomes. Adverum Biotechnologies is developing a gene therapy to treat the condition. Rather than addressing an underlying genetic cause, though, the gene therapy carries a sequence that causes the eye to produce aflibercept, the VEGF inhibitor marketed as Eyelea and Zaltrap. We spoke to Laurent Fischer, CEO of Adverum, about the company's gene therapy, how it works, and why the one-and-done approach could translate into better results for patients. Laurent, thanks for joining us.
Thank you for having me.
We're going to talk about wet age-related macular degeneration, Adverum, and its efforts to develop gene therapies for this and other conditions. Let's start with wet AMD. What is it? How does it manifest itself and progress?
So wet AMD, or wet or neovascular age-related macular degeneration, is a disease that affects millions of people globally. It manifests itself by fluid that accumulates in the macula and the retina, the site of vision. And patients who have this disease start losing their sight pretty rapidly. The current treatment for these patients, which has been developed since the mid-2000s, is to inject on a monthly or bi-monthly basis in their eye an antibody called an anti-VEGF. The leading drug is Eyelea or aflibercept. And it works quite well in studies where patients can actually recover part of their vision and maintain it with these frequent injections. However, in the real world, over a period of 2 years, for a number of reasons, patients tend to lose vision. So there's a clear unmet medical need for treatments that are longer lasting and can have better real world efficacy.
Well, what is the mechanism of action for a VEGF inhibitor? What's it actually doing?
So it blocks VEGF, which is a vascular endothelial growth factor, which is a reaction to the fact that the retina has a lack of oxygen. So it tries to develop new vessels. Unfortunately, these new vessels end up blocking the sight and the retina, the retinal cells, and blocking their function and interfere with vision. So they have a counterproductive effect. And by blocking that that VEGF with anti-VEGF, it actually reduces the amount of fluid and allows patients to recover their vision.
You mentioned that over time, the success rate isn't great with patients. How much of that has to do with compliance and the need to regularly administer this by injecting it directly into the eye?
Well, if you think about it, when you inject the anti-VEGF in the eye, you inject a significant amount, and over time, that anti-VEGF goes away. It's essentially reabsorbed in the circulation. And so, as the anti-VEGF goes away, the vessels grow back, the fluid accumulates in the macula or the retina, patients lose sight. And so some patients need more frequent injections every 4 weeks, some every 8 weeks. And you're correct. I mean, it's painful to have an injection in the eye. These patients need a caregiver to take them to their appointments. It normally takes a whole day, and they can't go about their lives. So there's definitely an issue with compliance that was very obvious during COVID-19, since their patients tend to be elderly and at risk. And in fact, there was a reduction in the sales of Eyelea in 2020 as a result of the pandemic. So this is absolutely a key factor in why in the real world we need longer lasting anti-VEGF.
Your lead experimental therapy is ADVM-022. This is not a conventional gene therapy in terms of correcting a mutated gene that underlies a disease, but actually produces the protein that's used in VEGF therapies today. What is ADVM-022 and what does it consist of?
So this is, you're absolutely correct. Most people think about gene therapies as advanced therapies that correct a defective gene and that are administered in patients with rare diseases. Here, what we've done is we've encoded the aflibercept transgene in our vector, which is injected inside the vitreum just like a standard of care in an outpatient setting. And that vector crosses what is known as the ILM, transduces the cell in the back of the eye to produce low levels of aflibercept. And what we've seen is that after one injection in our optic study, we had patients who had 109 injections prior to coming in the study. And over 2 years later, they're completely injection free and they've maintained their vision, and they have what we call dry maculas, which is essentially the absence of fluid buildup that we want to avoid in this patient population. What's unique about our gene therapy product, AV7M8, is that normally for ocular gene therapy, you need to actually have a surgical intervention when the surgeon injects the gene under the retina with a blob, and that has a number of issues and limitations. First of all, it requires a surgery, and the amount of virus you can put in that small space is limited, as well as the location and its ability to transduce enough cells to be successful. So ADVM-022 was actually evolved to be able to find its way through the ILM and transduce the cells with that simple outpatient in-office injection. And that's the result of direct evolution. It's essentially a platform we can use for other indications as well.
You need to get a patient to produce enough of this to have a therapeutic benefit. How difficult is getting the dosing right?
That's a really excellent question. And obviously, what you do is studies in animals first to see how much of that protein can be produced in the eye. We've done that in multiple studies and showed we can actually generate sufficient amount of that protein that is equivalent to what you would see roughly 4 to 6 weeks after an injection of Eyelea. And that's what you want to see as kind of the maintenance for these patients. And when we started our study, we started at a dose of 6E11, and we thought this would be the low dose. And what we found out is that 100% of the patients had 0 rescue injections. So all of these patients are now at 2 years and have had no need for new injections, which was really remarkable and unusual. And we dosed that down by 3-fold to 2E11. And what we've seen in these patients is that more than 2/3 of the patients had 0 rescue injections. And overall, if you look at the reduction in annualized anti-VEGF injections, it was an 85% to 96% reduction. So very dramatic improvements. And of course, in these patients, you can imagine you administer a vector with a transgene. You need to give them some prophylaxis to minimize any kind of immune response that we see, and we see some low level of inflammation that is managed with topical steroid eye drops. And based on these study, we're now planning to initiate global phase 3 trials that we plan to start later this year. And the hope is that we can file a BLA, submit an application for approval in 2024.
How localized is the effect of the gene therapy?
So the gene therapy, given with ADVM-022, with that 7m8 vector, is actually given inside the vitreum, and it actually broadly diffuses into the fovea, which is the localization of the sites and where all the quality of life kind of part of the vision are located. It broadly diffuses and transfects the cells and creates that biofactory approach where we see a very consistent, robust expression of that protein. So it's a really attractive way to have really a scalable solutions for these patients, because this is really will be the first mass market gene therapy product that potentially could reach over 9 million people globally if you include diabetic macular edema as well.
And what do we know about the durability to date?
So what we know from preclinical studies is that we see essentially lifelong expression out to 30 months in animals, and that's what you would expect from transducing post-mitotic cells. Those cells do not renew themselves, so they are unlikely to lose that transgene. And what we've seen in patients where we measured the protein in the eye of these patients is that the durability was beyond 2 years. We're going to continue patients out to 5 years, and that the protein expression was completely maintained out to that 2-year time point. Obviously, we'll continue to measure the protein expression level and continue to follow the patients. But the hope is that this will be essentially a lifelong treatment benefit. And this is in contrast to, the holy grail of anti-VEGF has been to extend the treatment interval from 4 to 8 weeks with current agents to 16 weeks. And now we're looking at potentially one and done. So a really transformational benefit for patients.
You're using novel vectors to carry the gene therapy. What does your platform allow you to do, and what properties do you consider in engineering a vector?
Absolutely. So that vector that we licensed from John Flannery's lab at UC Berkeley was designed using, it was rationally designed using direct evolution and testing millions of different vectors to identify the one that was suitable to be able to administer intravitreally or IVT, and could transverse that ILM, that membrane, to transduce the cells. And you want to transduce the cells in the back of the eye that are impacted by the disease. And that allows us to use that as a platform to carry other vectors, genes or proteins, to look at either rare disease or other ocular diseases. So this is a very unique capability of Adverum to essentially identify capsids that are very much targeted towards an organ or a cell, something that will allow us to build a pipeline behind ADVM-022.
So what might the phase 3 study look like? How large a population? How long a time period? And what are you using for endpoints?
Yes, so the clinical trial programs for anti-VEGF therapies are fairly well defined. And as you know, there are many agents that are either on the market or under review. And essentially what you're looking at is a 1-year-long study in treatment-naive patients that will receive either ADVM-022 at 2 doses or the standard of care Eyelea every 8 weeks. And the primary endpoint is non-inferiority based on vision, looking at a 4-letter. So you really need to show that your treatment is non-inferior to the standard of care. We'll continue, of course, to follow the patients to at least 2 years and potentially longer to see how long that benefit can last. But essentially a 1-year endpoint based on vision as the primary endpoint is what is required for all anti-VEGF treatment, including gene therapies.
One of the advantages of the approach is that benefit might not be limited to a single condition. You mentioned that you're also looking at diabetic macular edema. What is diabetic macular edema?
So it's a similar disease in the sense that there's fluid accumulating in the macula and the retina. So it has similar symptoms, but it's in the background of diabetes which, as we know, has a lot of impact on vessels and leaky vessels, and has can impact multiple vessels, and as we know. So in these patients, the presentation is much younger. It's slightly different than the age-related macular degeneration, but the treatment is the same. So essentially, these patients receive regular injection of anti-VEGF, Eyelea or Lucentis in the eye to control the disease. And we have a study ongoing called the Infinity Trial, where we're looking at, again, 2 doses of a DVM-022 compared to the standard of care, Eyelea. And this is the first actually gene therapy trial in DME. And that data will read out in the second half of this year. So potentially, we may be able to look at another indication for DVM-022 starting a phase 3 study sometime next year.
And how broadly do you think this ultimately might be?
Well, imagine that currently patients require these frequent injection in the eye by retina specialists. There are many parts of the world where either the cost of the therapy or the availability of specialists just does not allow patients to get treated, which means these patients lose sight and lose productivity and quality of life very rapidly. If you had a one-and-done treatment, we can follow patients, let's say, for 6 to 12 months using a simple in-office intravitreal injection. You could follow these patients even with new types of devices like at-home or CT to monitor the disease. And with a one-and-done, you could really scale this globally. In fact, we just hired Anand Reddy, who is going to head our patient advocacy, access, and digital innovation to see how we can make this treatment available globally, because we believe this is the first time we have a solution that could really scale up globally.
And is this an approach you might be able to take with other conditions, or is there something unique about the eye and VEGF that makes this possible?
I think certainly with the eye and anti-VEGF, we know that we're using a protein, aflibercept, that's been used in over 25 million eyes safely. So there's a strong foundation that we can use to do that. What we also know in the eyes that the doses of vector that require to be effective are around 10 to the 11. If you think about systemic gene therapy, it's much higher. It's about 4 to 5 log higher. And so this really provides an opportunity to look at a number of diseases in the eye and rare diseases. So we've been focusing in ophthalmology and rare disease for now, but we are also looking at ways where we could leverage our technology to target other organs like the brain or the kidney, and see if we can identify capsids that could actually be used for some diseases that are very specifically targeted to other organs.
Just want to go back to a point we talked about early in the discussion. You talked about how real-world outcomes don't match the clinical trial results with VEGF therapies. I'm wondering if you might expand on that a bit and talk about how your approach might change this.
Yes, so I think what we know from these real-world evidence studies that have been done both in the US and Europe is that patients do not get as many injections as they should in order to maintain vision. It ranges from over a period of 2 years, 5 injections in Italy to 6 in Germany, 9 in the UK, and 15 in the US. Despite that, patients are not getting enough anti-VEGF to control the disease. With ADVM-022, they get essentially a permanent expression in the eye. And so that's after a couple of weeks after they receive that injection, they start producing levels of anti-VEGF that are completely stable and hopefully lifelong. So the anticipation is that this will allow patients to preserve their visions for a lifetime.
Laurent Fischer, CEO of Adverum. Laurent, thanks so much for your time today.
Danny, thank you very much.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.